Good day to everyone joining us, and welcome to today's Xtalks webinar. Today's talk is entitled Overcoming Bioanalytical Challenges in Drug Discovery and Biomarker Identification. My name is Vicky, and I'll be your Xtalks host for today. Today's webinar will run for approximately 60 minutes, with time for a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this control panel. At this time, all participants are in a listen-only mode. Please note that this event will be recorded and made available for future download. At this point, I would like to thank KCAS Bioanalytical and Biomarker Services who helped develop the content for this presentation. They are a contract laboratory with more than 39 years of bioanalytical expertise. Centrally located in Kansas City, they provide small and large molecule PK immunogenicity and biomarker analysis, operating a variety of equipment platforms to service a wide range of therapeutic areas. KCAS's team leverages a highly scientific staff with an average tenure in the field of bioanalysis or biomarker analysis of 22 years to provide clients of all sizes with expertise in robust assay development, validation, and sample analysis under fit-for-purpose, non-GLP, GLP, and GCP conditions for discovery, preclinical, and clinical studies. Their teams have developed and validated more than 5,500 bioanalytical assays and have undergone 16 FDA inspections. And now I would like to introduce our speakers for today's event. Our first speaker is Dr. Jeanne Ray. Dr. Jeanne Ray is a senior scientific advisor at KCAS. He has expertise in a full range of bioanalytical techniques, including LC-MS-MS, GC-MS-MS, ELISA, RIA, protein binding, and mass balance investigations. Dr. Ray serves as a technical consultant and board member of a Phase 1 investigation review board. Jeanne has experience developing novel methods and has acted as the principal bioanalytical investigator. Jeanne frequently presents scientific findings at industry-specific conferences and previously served as the assistant director for numerous analytical chemistry programs for the National Cancer Institute while at Midwest Research Institute. And our next speaker for today is Mr. Laurence Goodwin. Mr. Goodwin is the Executive Director of Bioanalytical Services at KCAS. Business development, strategic planning, portfolio expansion, and client-based solutions are among his primary responsibilities. Mr. Goodwin brings with him the breadth of knowledge to provide scientific and technical leadership across the drug development spectrum, including drug discovery, safety assessment, and biologics testing solutions areas. He has more than 25 years experience in the life sciences industry and has authored or co-authored more than 35 papers and presentations and provided over 150 internal documents. Laurence is an active member of American Society for Mass Spectrometry and a current member of the executive leadership team at KCAS. And now, without further ado, I would like to hand the mic over to our speakers. They may begin when ready.
Thank you. Hi, this is Jeanne. The presentations for today will include the following topics: unstable compounds, complex tissues, large and divergent analysis and dynamic ranges, non-specific binding, the time constraints on discovery, and limited sample size.
So in the bioanalytical field, what are some challenges your clients have faced when working with unstable compounds?
That's a good question, Vicki. We will talk about matrix evaluation first, as there's a variety of matrices that come into play here. We'll talk about tissues mostly later on in another slide, but right now we'll talk about plasma, whole blood, and other matrices. really it comes down to the analyte of interest. You may have some higher recovery of the analyte in certain matrices over others. This can also be inclusive of biomarkers, which we wanted to touch base upon as well today. Some of the key focuses of that would be the instability of some of these analytes in certain matrices over others. So you really have to do an evaluation to see which matrices is going to provide the best solution for you to be able to analyze the compound of interest and be able to develop a quantitative assay that is robust and reliable. Talking about robust and reliable, some of the things that come up are stabilization of the compounds that are of interest. So we here at KCAS have a long list of 6 things that we look for. Stabilizers that we can add, which will eliminate any esterase activity. Also, we look at rapid processing of the matrix to plasma, potentially to remove any kind of hemolytic effect that may occur or any congealing of the blood. So we understand the importance of the criticality of the timing and the exact pipetting. We also will stabilize some compounds by acidifying them. And this can be done with a variety of acids, including potassium oxalate as well. But you have to really be careful with that because the pH is crucial, and sometimes you'll get some gelling or some coagulation of the blood or congealing of the plasma. And you also want to avoid any esterification of alcohols. Now, cold storage and handling is a very critical step. Any cold centrifuges for sample processing from whole blood to plasma would be beneficial, as well as any immediate freezing of the samples once processed, not letting them sit around at room temperature. We would prefer that all of this work be done on an ice bath to help alleviate any of the room temperature conditions that may cause destabilization of the compounds. One of the things that we have a lot of value here is derivatization. So we have an example, if we had a free thiol that was linked to a prodrug, we would have to cleave it and then we would modify it or derivatize it with, let's say, NEM, for example. So these are some of the things and steps that we can take and precautions that we look out for. Having the information of the compound really helps out as far as structure, functional moieties, molecular weight, and the type of compound that it is. So we also protect it from light if it is photosensitive. That could be either yellow light or red light, depending on the sensitivity of the compound. So back to biomarkers. And I'll let Jeanne talk about sample collection protocols here in a second, but I want to touch base on biomarkers specifically. There's really 3 ways that we can analyze those. The first way that most people would do would be a neat solution curve, a neat calibration curve. That really doesn't take into effect the matrix suppression or enhancement or recovery that you would get from having that matrix present. The second way would be a surrogate matrix that is closely related to the biomarker, but might not be exact. So this could be plasma that was stripped with charcoal to remove any of the endogenous biomarker present so that you can analyze it in that curve. But the real approach and the old school approach is standard addition, where you analyze the compound by adding a known amount of the reference standard into the actual sample unknown, and then you can back calculate to determine what the concentration is. So these are some of the approaches that we can take. But speaking of matrix suppression enhancement, now that's a change in the response of the target analyte as a result of the presence of interference in the sample. There are some ways that we can avoid that neat versus matrix optimization. We would prefer to do matrix optimization as opposed to neat, so you actually can see the matrix effect, whether it's suppressed or enhanced. Post-column matrix infusion can be performed to determine selectivity of lots of plasma or whole blood, and using APCI over ESI, eliminating the plume or electrospray and going into a gaseous phase. That eliminates most matrix effects. But still, that may not be enough to overcome that, so we would look at column technologies, potentially trying to separate out the interferences from the analyte of interest. And then we would always recommend a stable isotope label IS to help at least minimize or eliminate those matrix effects, because that IS should behave as similar to the analyte as possible. So Jeanne, if you want to talk about sample collection protocols.
Yeah, I can't underestimate how important to have an appropriate sample collection protocol. I want to share one example that we were faced here recently. It was a low molecular weight aldehyde that we were tracking in dog blood. It undergoes extensive degradation by 4 separate isozymes, and the half-life of the drug was approximately 15 seconds in whole blood. After extensive evaluation, our team found that the only effective way to stabilize the drug was volumetric additions of ice-cold mixture of acetonitrile and acetone directly to the whole blood. The addition of the acetonitrile, which you would normally use, was just not sufficient alone for stabilizing the compound. So the challenge was then to create a workable sample collection protocol for the non-clinical site to implement in sample collection. We've successfully now completed the GLP phase of this program, and the drug is now entering clinical trials. So we then had to further modify the sample collection protocols for bedside collection. So here is an important aspect that it has to be reasonable, and it has to have ability to be compliant for sample collections and have the integrity of the sample ensured.
Okay. So talking about the stabilization, adding components that can stabilize the compound, including esterase inhibitors, antioxidants, or enzyme inhibitors. You know, you would do this in whole blood in situ, and that'll help stabilize the compound. You process it from whole blood immediately to plasma on ice to eliminate any kind of temperature effects. This can go as well in tissues where you would ice cold methanol or ice cold solvent system or buffer to do homogenization with, which we can talk about the tissues a little later. And then one of the things that we do here for very small compounds that are very polar, that are unstable, we can help stabilize them in whole blood by performing derivatization in the whole blood directly. And having those kits supplied would really be beneficial in that, because the exacting amount in the pipetting is very important.
So can you explain how you have overcome the complexities related to developing bioanalytical methods in tissues?
Sure. I want to focus on some more difficult tissues, and these are the hard tissues, typically bone and nail. These are challenging matrices to work with. We found that the addition of a basic media or extraction buffer has been very effective in softening the tissue prior to homogenization with a bead homogenizer, such as the Priscellas. Examples that we have found effective in this arena is terbinafine in nail, and the bisphosphonates as a recidronate in bone tissue. Other tissues that are very difficult for laboratories to work with are those that tend to congeal once homogenized, and that is lung, skin, and gastrointestinal tissue. As difficult as hard tissues to work with, these specific tissues can be very difficult to work with. When they're homogenized, they tend to recongeal into a ball, resulting in a heterogeneous sample, difficult to pipette, and all the issues related to compliance for the assay. We found that the addition of an acidic buffer often softens the tissues to an extent that when homogenation occurs, the sample maintains its homogeneous condition. Other things that are problematic for labs are samples that are contaminated or stratified. So an example would be cerebrospinal fluid, where it gets often contaminated with blood, resulting in much higher levels than should be expected. And sputum can be very problematic in the fact that it often is stratified with variable drug concentrations. Sputum can be really a difficult matrix when you're looking at an inhalation drug administration, where the drug will be very highly concentrated in part of the tissue and very low concentration in other sputum samples that are collected. So it's important that when you're looking at a sample collection protocol, as well as in how it's processed, these samples for like sputum have to be completely homogenized, as well as feces, so that you're not looking at the stratified samples. And when a study then moves forward into a GLP compliant, you can't be working with a portion of the tissue. You need to work with the entire tissue. Otherwise, your incurred sample reanalysis will tend to fail and not meet acceptance criteria. Other things that are helpful is instrument optimization. We found that column and valve switching can often be effective, eliminating background and matrix interferences from complex tissues. And lastly, customization of extraction solvents, buffers, and inhibitors, as Laurence said, can be very important, not only in the extraction efficiency, but also maintain the integrity of the analyte to meet the assay sensitivity requirements, and modification of the sample extraction procedures needs to be considered at that point in time.
So I'd also like to mention on that slide there, this applies to biomarkers as well. And so typically in the PD biomarker study, you're looking at either kynurenine or some other amino acid or some other metabolome that may be present that you want to look at. You really want to measure the upregulation or the downregulation as another drug is dosed to the animal. And so a lot of those take place in muscle tissue. They also take place in kidney or liver or sartorius. So there's a lot of places that really come into play when you're dealing with biomarkers, because biomarkers tend to express themselves in certain areas throughout the body differently. So every matrix will be different when it comes to that. So that's a point that you really need to take into consideration when you're developing a PD biomarker study. Okay.
In your experience, what has been the best way to establish the analytical range to support a discovery assay?
That goes to being in discovery. One of the things you would want to do is have a very large dynamic range. We found that not having any information up front on the molecule, if it's first time in animal, or if, sorry about that, I forgot to push the slide. So not having any information on that, as we all know with the LC mass spec, the curve tends to bend over and the dynamic range is only 3 to 3.5 orders of magnitude. Sometimes you can get to 4. But if you expand that range and go to 5 or 6 orders of magnitude, then you're able to have a low curve and a high curve together where you can actually determine what fits the data better. So by doing this, if we tend to run a semi-log curve from, let's say, 0.316 to 3160, that's a 5 order magnitude curve, or from 0.1 to 10,000 nanogram per mil, and that would be a 6 order magnitude curve. We then would run the assay with those curves in place, and the samples that would be analyzed then would fall in a range. They can either fall in the middle, which would be perfect, or they can fall on the low end or the high end. But by doing that, having that flexibility of that curve and having it set up, we can truncate the curve to fit the data, meaning that the range of the assay, it could be anywhere from 1 to 1,000, or from 1 to 10,000, or from 0.3 to 3160. So by having that range truncated, we actually fit the data to the curve, which means we're analyzing for that specific data and make it fit for purpose. And it allows us the flexibility to get the most data from the sample sets without having what we call BQLs or AQLs, below quantitation limits or above quantitation limits. Now, that said, you will sometimes have something that's dosed extremely high or has great sensitivity, and it may be above the quantitation limit. But what will happen is then we have the flexibility with the control blanks that will do a post-extraction dilution with the control blanks that have already undergone the extraction process with the IS present. And so by doing that, we're diluting like with like, and then we can actually run it again against the same curve and then have that data at that much faster pace. So the large dynamic range does help. You would have disparate levels for metabolites as well, which tend to be typically lower, analyzed in a single batch. And so what will happen at that point is you can run 2 different sets of curve by having this large dynamic range. You can have the lower end set up for the metabolite and the higher end set up for the actual analyte of interest. So by performing a single extraction and having both of those transitions for the metabolite and the analyte present, you would be able to analyze those in a single batch, which increases your efficiency to be able to get results for everything while having a robust or a fit-for-purpose assay. So one of the tricks that we utilize to get the most information from that curve is we would set up multiple MRM transitions. The first one optimized for lower curve range, and the second parent-daughter optimized for the higher curve range. And this allows us to get a better linear fit overall for the data that we're going to collect.
What are some steps you have taken to address challenges encountered when collecting samples?
Sure. I think probably one of the most problematic for laboratories is when you're working with cerebral spinal fluid or urine. These are low protein matrices. Drugs in particular in cerebral spinal fluid can be lost to the sample container due to what we refer to as non-specific binding. This is particularly problematic for urine samples when they're collected over a given time interval, for instance, a 0 to 4 hour period, and the aliquot is sent to the lab for analysis. Significant portions of the analyte may be lost to the sample container, original sample container alone, and the portion that's shipped to lab may not be reflective of the actual sample that was collected to begin with. The sample container evaluation, we look at a wide range of containers, including polypropylene, polyethylene, polycarbonate, glass, and even silicized glass can be options for sample collections at the clinic or the non-clinical side. So from a laboratory perspective, we do what we call a simple 5-tube evaluation. This is a simple test. We take the desired matrix, we'll put it with the test compound and transfer aliquots from the original tube to 4 sequential tubes in succession, which increases the sample surface area exposure in each of the steps. The level of the test compound in the 4 tubes are then compared to the original tube. And if there's a concern that the drug may be binding to plastic to begin with, a separate portion would be placed into a glass container as a control. Not to be overlooked is to look at the transfer pipette that's going to be used in your sample collection protocol, because this can result in significant loss of absorption based on what the lab may be using. And also, consider the vacutainer blood collection tube type. Nitroglycerin, for example, needs to be collected into a glass vacutainer tube, not a plastic tube, which is usually more of a standard fare in clinical applications. And lastly, I do want to mention that clinical kits are an important aspect of what laboratories do provide, and KCAS does have a blog on this particular topic, which shows the importance of putting together the kit that's reflective of the sample collection protocol.
How have you dealt with rapid turnaround times related to the access to data to meet client needs?
So one of the approaches we take here is integrated drug discovery approach that includes a tiered research assay that we develop, which falls into a reverse phase paradigm. This can be for small molecules, peptides, biomarkers, any kind of typical small molecule or atypical small molecule that we would analyze on an LC-MS system. So this tiered approach consists of standardized methods with generic ISs and column technologies, which can include polar NCAP C18 columns, C8 columns, UPLC, HPLC columns, so 1.7 micron or 3 micron or 5 micron. We typically tend to utilize smaller column technologies, 2.1 by 50s or 2.1 by 30s. The reason being is that allows us to get a more rapid injection to injection time so that we can have samples ran in less than 5 minutes per injection so that we can do a batch relatively quickly to eliminate any issues with stability or any issues with taking up too much time on the instrument overnight so that we can have it done by morning. Some of the things in our toolbox include for this standardization would be utilizing protein precipitation for typical small molecules. That tends to work for 75 to 80% of the analytes that fall in that category. Another option would be liquid-liquid or solid phase if it was more advanced and required a little bit more cleanup, or if it was endogenous. Having multiple generic internal standards, we include one that is a polar, which will have less than a 4K prime. We include one that is moderate IS, which would be 4 to 8K prime. And then we have a non-polar IS that is greater than 8K prime. So what this allows us to do is we're able to have these diagnostic probes present throughout the column technology so that wherever the analyte falls, we have an IS that we can utilize to generate the peak area ratio from that is more consistent and would be better served as an IS for that typical compound. So one of the things with rapid turnaround time is if we want to do this approach in discovery mode, we want to have 3 to 5 day turnaround time. So in order to do that, you have to bring it in, you have to have standardized methods in place that allow you to have the flexibility to get the data out the door. And so we have that here. We have peptides mentioned up there. That's a special case because you have to really be careful with peptides depending how large they are. They may crash out if you do a protein precipitation. So what happens there is we screen multiple extraction solvent systems to determine the best fit. And then we also can perform a pellet recovery test to determine how much of it did crash out if we are performing a protein precipitation. This typically works for most peptides or smaller proteins between 2 kilodalton to 15 kilodalton. And then we would use a multiply charged data to measure it intact. If we wanted to do larger peptides or proteins, we would have to undergo digestion and analyze, which the analysis would be performed around a signature peptide that we've identified that's unique to that particular protein or peptide. But the tiered approach does allow us fit for purpose, qualified methods and validated methods. So the more robust the method gets, the longer it does take. But with the early discovery, that would include the high throughput PK, that turnaround time would be 3 to 5 days. The lead optimization, which would be a little bit more robust, would be 5 to 7 days, where we actually spend 2 days of method development optimization on extraction technique, extraction recovery, extraction efficiency, and also mass spec optimization as well as LC optimization. And then, of course, if you get to pre-IND, that's the same criteria as it would be for a GLP, and you can qualify that or validate that as the needed fits for the sponsor. We're flexible in that regard. Those would be typically for candidate selection. So as you move through the drug discovery process, you go from hundreds of compounds that you want to screen to tens of compounds that you want to optimize to a handful of compounds that you want to send towards IND and get ready for preclinical trials by GLP. That's all I have there for Jeanne.
What solutions have you developed when dealing with limited sample size in the past?
Sure. I think the key for us is looking at something called microsampling, and that could be involved with human tears, rat milk, for example, and with microtissue sampling that we may receive for instance, rabbit retina, rat urethra, or even dog earwax, which we're currently working with, just naming a few, each with just a few milligrams of tissue that may be provided. So in each of these cases, for capillary sampling or microsampling, it's important that the entire capillary be transferred to an appropriate tube that can be used directly for sample extraction for the laboratory. That's something that's been provided in the sample collection protocol. In the case for micro tissue samples, it's key that the sample be transferred to a tiered sample container where the weight is recorded for the laboratory. Typically for us, we like using something like the microtube provided by Priscilla for bead homogenization, and having that already in place makes it so much more reliable for the laboratory to process the sample in an efficient manner. And it goes without saying, I guess, but with any microsampling, optimization of both the extraction efficiency and minimizing the reconstitution is essential for achieving the desired assay sensitivities. But further, you may be looking at optimizing analyte detection, and key what might be of value for the laboratory is to looking at a derivatization which can enhance the detection of the analyte in the mass spec. This can also not only improve the molecular weight of it so that you can separate it from endogenous components and you have a much better signal to noise. Microflow LCMS is also another option. It has some advantages in the fact it will improve peak shape and therefore detection of signal to noise.
So if you could back up 1 second, I can talk about a little bit about optimizing analyte detection, Jeanne. So as far as derivatization goes, this would be very good for compounds that don't have any functional moieties that ionize very readily on a mass spec. And so these could be steroids, these can be very nonpolar compounds that have an embedded amine that's hidden so that it's a tertiary amine in a ring structure that's not going to light up very well. So we can derivatize it with an NEM or some other ester if there's a OH that's present to help light up the mass spec, if you will. With the microflow LCMS and also with nanoflow LCMS, which is more prevalent now for proteins, which we will be moving into that field here shortly at KCAS, looking at optimizing that flow rate with the increase in detection of the signature peptide for proteins. So those become critical. The lower flow you go, the more sensitivity you can get on the mass spec based upon either the ESI or APCI sources that we would utilize. But it becomes very critical to optimize your analyte detection, and that goes back to the large order of magnitude range we would use. We would optimize our detection for the lower end of the curve, but we will also, a term that we utilize is de-optimization on the higher end. If you have a lot of sensitivity with the compound, you're going to have to de-optimize that to get a larger dynamic range. And so really the optimization and de-optimization in the mass spec becomes very critical at that point. Thank you.
Well, thank you very much for that insightful presentation. And before we get to that Q&A portion of the webinar, we do have a quick poll question for the audience, and that should be launching on your screen right now. And that poll question is asking, what areas have you found to be the most challenging in drug discovery and biomarker identification? So if the audience could please get their answers in, this poll question will be closing shortly. And the options we have here are unstable compounds, complex tissues, large or divergent analytical ranges, time constraints on discovery, or limited sample size. And once again, if the audience could get their answers in, this poll will be closing shortly, and we will be moving on to the Q&A portion of this webinar. And once again, this question is, what areas have you found to be the most challenging in drug discovery and biomarker identification? And it does look like majority of the audience has voted, so I'll go ahead and close this poll question and share the results with everyone. And it looks like majority of the audience with 38% voted for unstable compounds. After that, it was 23% with limited sample size, and then a tie of 15% with large or divergent analytical ranges and time constraints on discovery. And lastly, 8% of the audience voted for complex tissues. So Laurence, Jeanne, any thoughts on this poll?
Go ahead, Jeanne.
No, yeah, I think what's most important is looking at how to best stabilize unstable compounds. This can be so problematic for a laboratory and knowing how to best approach it. We're not only looking just for the parent compound, but also be looking at key metabolites that may be also labile in the assay. So for the laboratory to have a clear understanding of what's the best way to stabilize it, have the proper sample collection procedures, because once the sample is collected, that study basically has launched and you're not able to retract the data back from that. So.
Yeah, so my thought on that is it's exactly where we thought it would be with unstable compounds, because those tend to be the most difficult, and a lot of shops that are other contract research organizations like us do not have the experience to be able to handle those type of compounds. You know, this could be something as simple as adding dichlorvos to the whole blood, or E-serine or acetylcholine or other common esterase inhibitors, but it might be as crucial and critical as derivatizing, where you spike, store, hydrolyze, and derivatize a thioester prodrug immediately so that you can stabilize it so that it doesn't break down or it doesn't form any more dimerization as most free thiols will do, to something as simple as the amount of time that's processed, so that you really have to educate the clinical sites when they're collecting the samples that time is a critical function, that they need to process within 30 minutes. They need to keep it on cold ice. They need to use a refrigerated centrifuge, and then they need to deep freeze it as soon as possible in -20, -70, or -80, depending on the criticality of the.
And flash freezing in particular can be a really important aspect of
tone
it.
bath, yes.
Such as in ADCs.
Especially with ADCs, where the linkers can break, and then you're introducing more free than total. Yep.
Perfect. Thank you so much for that insight. And now I would like to invite our audience to continue sending in their questions or comments right now using the questions window for the Q&A portion of this webinar. I've already received some questions, so I'll go ahead and get started with those. And the first question we have here is for Mr. Goodwin, and that question is, what SPE plate do you suggest to try first for peptides?
So there are a couple SPE plates that are pretty standard. One of them is the Waters Oasis, which tends to work, but now Thermo has introduced a new plate that is fritless, which is really kind of neat. The actual sorbent has been homogenized with the frit, so and then polymerized, so it forms a very very unique way to look at an SP. It almost looks like an old number 2 pencil rubber eraser, and that tends to not get clogged up by some of the larger proteins or some of the things that you may have to use with the digestion if you're going to analyze it. So we found that works very well from Thermo Scientific. There are some sorbents out there that tend to have a larger pore size and a larger angstrom. So I would tend to stick with 230 angstrom or 300 angstrom sizes on those. That will help trap your protein in your peptide of innate interest, so that it doesn't elute directly through in the wash step.
Great, thank you for that response, Mr. Goodwin. The second question we have here is also for yourself, and that question is, can you please describe the post-analysis dilution technique?
Sure. So if you have your plate of standards and QCs, if you have them at that level and your samples, you also have control blanks that you run as well that contain the IS that everything contains except for the true double blank or the matrix blank. So having undergone the extraction process, you then have analyzed everything. You know that your control blanks are blank and that there's no interference there, and you only have IS present that matches the rest of the run. You can thereby take and use those to pull together and use those as a dilution to dilute the AQL samples to the appropriate amount to get that back within the curve. And then what we do is we take it one step further. Not only we dilute those samples because we're running it against the same batch the next morning, we will take one of the medium standards and dilute it as well so that we show that the dilution works, and we use that one as a QC.
Great, thank you for that response. The next question we have here is, can you assist us with clinical kits associated with collecting unstable compounds?
Yeah, this is Jeanne. So we provide that as a core line of business for our sponsors. We typically send out about 3,000 kits on a given month, and these do include providing a stabilizer within the sample collection tube itself. We also will provide the stabilizer. It can be in powder form that can be reconstituted in the lab where there's limited stability of the stabilizer once reconstituted and applied to the matrix sample. So whatever the given need is for a particular study, we do provide. It can be something as simple as providing calibrated pipetters. We've had studies where the clinical sites don't have that expertise, don't have the documentation that their pipettes are even calibrated. So we will send those calibrated pipettes out in the kits. Then they are shipped back to us, and we recalibrate them and show that they were in compliance during the sample collection intervals for that given number of subjects or parts of the trial.
Yeah, I'd like to add one more thing to that, especially if you're going to deal with any kind of derivatization. I would recommend that you would get a kit. Not just from us, but whoever you're going to use it from. You're going to need that expertise that it's going to have to be pipetted exactly. You're going to have all the different tubes that are associated with how you're going to perform that derivatization, and then the quenching, and then the freezing. So I would recommend that a kit be utilized in that case that is customized.
Perfect, thank you. The next question we have here is for Mr. Goodwin, and that question is, what is your default ionization technique?
So the default ionization technique is either going to be ESI or APCI. We have the flexibility to do both. If we know the structure of the compound, we would default with APCI. Typically, if we did not know the structure, if we know the structure and we know that it's going to be not sensitive, then we would move to ESI. ESI tends to be an order of magnitude more sensitive than APCI, but APCI tends to eliminate matrix effects, and this way you don't have to deal with any of that suppressant or enhancement due to matrix effects.
And we may also look at both positive and negative ionization modes. Negative tends to be less sensitive than positive, but we will evaluate both.
Yep.
For the next question, this audience member writes in, can Mr. Goodwin comment on the different levels of assay and what level he would recommend for discovery?
So I would recommend using our tiered approach. We have RGA1, which is for early discovery. High throughput PK would fall into that category. For RGA2, which is research grade assay 2, which is a little step up, it's a little bit more sensitive. The accuracy and precision on that one would be 20% with the LLQ at 25. That is a little bit more, it's still fit for purpose, but it has a couple days of method development so that we can really make a more robust method. And then the third approach on the tiered approach is for the pre-IND, and that would be RGA3, where it's plus or minus 15% with 20% LLQ, which matches what you would be going into preclinical or clinical validation at 15 and 20%. And the time there, which is a nice thing about the way this is set up, going from early discovery to lead optimization to pre-IND to clinical, or preclinical to clinical, is as you go down, when you get to RGA3, you're at the same level. Those method development days there, which would tend to be 3 to 5, will be counted towards any validation method development days, because we're going to use the same techniques, we're looking at the same thing, and so we have a process in place that kind of guides you through wherever you're at in your integrated drug discovery approach. We have the assay that helps fit your needs.
Perfect, thank you. Another question here for Mr. Goodwin, and this question is, what % accuracy criteria is typically used for peptides in discovery?
So right now, there's a debate going on as whether or not that should be an extra 10%. We find that we're able to hit the criteria that we have set up in discovery, which is 25% with LLQ at 30%. But if the client knows that it's going to be very difficult, we will inform them that we may want to open that up to 35%. So with peptides and endogenous biomarkers and with some proteins, you know, you've got the biotherapeutic piece that plays into that we tend to be able to hit here the same criteria as a small molecule, but normally it isn't treated as a small molecule. They're treated like a large molecule, which tends to have slightly looser criteria. So hopefully that answers the question. It's going to be a case-by-case basis, really.
Agreed.
Great, thank you for that response. The next question we have here is, do you typically evaluate matrix effects or suggest a client to add this test in for your early phase discovery bioanalytical methods?
So for RGA2, we would evaluate matrix effects and matrix suppression. That's why we add in the generic IS cocktail, if you will, so that we can have one IS that is polar, one that's not so polar, and one that is non-polar. And we use the Goldilocks theory of wherever our analyte elutes, that's the IS we want to really use with it. And so that helps us evaluate the matrix effects. But as you move into an RGA3, we would recommend that you use a stable isotope label to help minimize those matrix effects. But it is always evaluated in an RGA2 or 3, but not so much for high throughput PK, early discovery. You're just running a curve, and it's a fit for purpose, and the data is truncated to fit all of the samples, or the curve is truncated to fit all of the samples. So it really is not necessary at that point, but it is something to keep in the back of your mind. We recommend as you move up to RGA2 that you look at matrix effect and you look at benchtop stability.
Perfect.
In addition to performing the analyses.
Perfect. The next question we have here is for Mr. Goodwin, and that question is, what is the data turnaround time for non-GLP projects from sample receipt?
3 to 5 days for early discovery, 5 to 7 days for lead optimization, which would be RGA1 and RGA2 respectively, and 7 to 10 days for RGA3, where there's 5 days of method development. So, you know, if you do 2 to 3 days of method development for RGA2, that adds to the 3 to 5 days, but we usually can get that turnaround in 5 to 7. And then for an RGA3, if you had the 5 days, we typically get that turnaround with 7 to 10 days.
And the next question we have here is asking, why have a tiered approach to drug discovery studies?
Well, yeah, I think the intent here is to have a fit-for-purpose approach. The RGA1 level, which is basically sort of a down and dirty approach. It gives a very cost-effective, timely response when somebody is looking to see whether or not the drug has particular pharmacokinetics that it needs, the right half-life, that it maybe has a biomarker that they're also looking at the same time, which shows the efficacy that they're looking at. And as you move further into what the RGA2 level is, that's a qualified method. And again, as Laurence was saying, there's some degree of stability that's established, more of a matrix effect evaluation. And by that, we look at hemolytic, we look at lipemic, and we look at other lots within that matrix to make sure that there's not going to be a problem for the assay. And then as you move further into the RGA3, which is a more, if you want to look at it, fully validated, qualified assay, that has a lot more stability to support the meaningful stability that you can have a reliable data result from that.
Yep, and the big thing with RGA3 is it's what I like to call GLP light, meaning that we're running it just like we would a GLP method development in qualification or validation, except for the QA process is missing.
That's correct.
So, but as you go across the tiers, the reason they have the tiers is you don't need to hit everything with a sledgehammer. You need to, you know, if you do that, then you're overpaying, overcost, it takes too long. So in early discovery, you really need fast and efficient to get you your answer so you can rank prioritize your compounds of interest, determine which ones have the efficacy that you want, as Jeanne mentioned, to move forward to the next stage. And that's where you spend a little bit more time and you might get out a ball peen hammer instead of the big sledgehammer, which is GLP. Because then now you're talking about, as we mentioned, the days go from 3 to 5 to 5 to 7 to 7 to 10 to a month in GLP. One week of method dev, 1 to 2 weeks of validation, and 1 to 2 weeks to write the report. So it really depends. You know, we know that discovery has to be fast, flexible, and nimble. So by having this approach, we're able to give our sponsors what they really need to make the decisions to move forward, to move their drugs from left to right in their pipeline.
Great, thank you for that response. The next question here is, what are some of the stabilizing regions for unstable compounds?
Okay, so for a wide range of stabilizers that may be used, you may be looking at DFP, which is very toxic, or we can also look at dichlorobos, as Laurence had mentioned earlier. For esterates, we use sodium fluoride potassium oxalate as an inhibitor, particularly in the blood collection tube. These are very effective. You're also looking at adding reducing agents to keep the dimers from forming. The DTT, it could be NEM as part of that derivatization process. So each compound is unique. Each compound has unique aspects. Sometimes the derivatization does require some degree of heating or time constraints. So stability could be an issue. So each of these really are on a case-by-case basis. And as Laurence mentioned earlier as well, light protection is also important in this process for stabilizing during this process as well, not only at the clinic, but also in the laboratory as well.
Yep.
Perfect. And the next question we have here is for Mr. Goodwin. And that question is, how is drug-to-drug contribution monitored in a multi-analyte discovery LC-MS-MS assay?
Okay, so one of the things you could do there, if you have multiple drugs in the assay, which could include metabolites as well, is you would look for crosstalk initially to make sure that there's no interference or no peak rising from just analyzing them individually. So when we're doing the optimization for those, we would look at them individually with all the MRM channels present in the assay to show that it's only being detected on the channel that it's supposed to be detected on. And so that eliminates the crosstalk effect. So then you also can use your column technologies to help you separate those out to the next degree, which is the second dimension. Not only do you have the MRM, but then you have the chromatography so that they are separated out. And then as far as looking at drug-drug interaction, if you want to go to that step, you would have them in the presence of these other drugs and without, and then measure to see if there was a loss of signal or an enhancement of signal with the other drugs present. So those are the three key things that we would look at when evaluating for drug-drug crosstalk, drug-drug interaction, or drug-drug influence.
And importantly, look at cassette dosing because they're all very similar in structure and can be co-eluting.
Yep.
Perfect. Thank you for that response. And this brings us to our last question, and that is, can you provide data on cassette-dosed compounds?
Oh,
yes,
we
can. We can do 5 or 6 in a high throughput PK mode, and we typically do a lot of GLP studies where we're looking at not only the parent compound, but multiple metabolites, as well as some derivatized metabolites and derivatized compounds. So we can look at up to, the mass spec will be able to do anywhere from 11 to 30 MRM transitions pretty consistently. We'll have the bandwidth and the counts per second on the peak to be able to determine that, because you'll have to do switching. It's a little bit more complicated when you have to set up 2 periods, if you have to do a multi-state where you have a positive and a negative, but it is still doable. You just have to account for all the transitions and in between, because each transition you look at costs you 5 milliseconds of time while the mass spec refocuses on that new transition. But it is possible. We typically can do high throughput PK screening on 5 to 6 compounds that are cassette dosed in an animal with a 3 to 5 day turnaround time.
Perfect. Thank you for that response. And so Dr. Ray and Mr. Goodwin, any last thoughts before we wrap up today's webinar?
I guess from my perspective, what's key is the fact that a sample collection protocol is key. If you don't know how to collect the samples, if the laboratory hasn't properly designed it so that it is a practical sample collection procedure, it's basically deemed to fail. So keeping it simple, stupid is an appropriate thing to consider when you're looking at a sample collection protocol, especially moving to clinical, because you're looking at multi-sites, multiple levels of expertise. A non-clinical site tends to be much more adaptable and able to comply with that, but not when you're moving to clinical. I think that's where we see the disconnect.
Yeah, I agree with that. The one take-home message that I have is be able to share information around the structure if you're the client. The more information that you arm us with, the more tools that we have at our disposal to be able to give you the answers you need and partner with you to really get you the data that you really want and desire. So, you don't necessarily have to share structure, but if you share elemental formula, the free base molecular weight, solubility, any physical chemical properties of the analyte, such as log P or log D, pH or pH that it is unstable in, any of these things help. All that information that we have allows us to be able to develop a much better assay for you, instead of trying to guess and then pull out a bunch of tools and that you're going to have to pay for, but you might have already had the information to give to us.
Well, thank you very much for those answers and those thoughts, and we've reached the end of the Q&A portion for this webinar. If you do have any further questions, please direct them to the email address showing on your screen, or if you would like some more information, please visit KCAS's blog for more information. And thank you everyone for participating in today's conference. You will be receiving a follow-up email from Xtalks with access to the recorded archive for this event. A survey window will be popping up on your screen, and your participation is greatly appreciated as it will help to improve our future webinars. Now, please join us in thanking our speakers, Dr. Jeanne Ray and Mr. Laurence Goodwin, and you can do so by clicking the link in the chat box. We hope you have found this conference informative. Have a great day, everyone.